I'd like to thank you all for participating with us tonight. I know it's quite late for some of you, maybe even quite early in the morning for others. For those of you whom I've never conversed with, my name is Dr. Eric Kusher, president of SCORE Medical. SCORE is a company which focuses on science-based, clinically proven skincare products and services exclusively for the professional community. Tonight in particular, we're going to be focusing on a specific suite of procedures for the physician only, developed and offered through our partner company, the Ageless Regenerative Institute. These techniques and products are designed to offer the physician a comprehensive in-office approach to extract, harvest, and process stem cells from the patient's own adipose tissue or platelet-rich plasma for reintroduction into the skin for long-lasting, effective cosmetic regenerative effects. We should be together for about 30 to 45 minutes. Without further ado then, allow me to introduce you to Kristen Comella, the Chief Scientific Officer of ARI, who's going to guide us through a brief presentation and an overview of the system, followed by a Q&A session, and finally, I'll come back to you at closing with details on how you can obtain full information on scheduling and participation if you're interested in pursuing the educational and practice-building opportunities. Kristen?
Thank you, Dr. Kusher. Thank you, everyone, for joining us and taking the opportunity to learn a little bit about stem cells and how they can be utilized in true regenerative medicine. Thinking about the Ageless Regenerative Institute, we are dedicated to standardizing and elevating the field of regenerative medicine through research and medical education. Ageless as a whole umbrella has been around for more than 10 years and has focused on educating physicians on a variety of different opportunities and techniques that can be used in the clinic today. We're collaborative with academic centers in the U.S. and researchers worldwide, and we have a faculty of subspecialists that are part of our community. The purpose of this presentation is to offer physicians guidance in a proven, validated, compliant outpatient method to provide both platelet-rich plasma as well as adipose-derived stem cell injections into their offices. So let's talk a little bit about autologous stem cells. So these are the stem cells that come from your own body, and in particular, I'd like to focus on something called adult stem cells. I think there is a lot of confusion out there about stem cells, and the general community and general public tend to think of embryonic stem cells when they hear the word stem cells, and they forget that there are, in fact, stem cells that are part of every one of us. And these are the adult stem cells that we're referring to. These are found in many different tissues, and their main purpose is to maintain and repair the tissue in which they are found. They can be found in both an undifferentiated type state or a differentiated state within those tissue samples. The thing that's unique about the adult stem cells is that they are multi-potential, which means that they have the ability to differentiate into a subset of cell types, and they are not pluripotential. In other words, they cannot give rise to a full human being or baby. They are multipotential and can give rise to every tissue within an existing body. The other thing that's unique about adult stem cells is they may exhibit something called plasticity. What this means is that they're easily influenced by their environment. Whatever you put them into, they tend to form that. So in other words, if you place them into bone, they tend to form bone. If you place them into muscle, they tend to form muscle. So I'd like to talk a little bit about adipose-derived stem cells, which are a specific type of adult stem cell which is found in the fat tissue. These cells that are within there are different from blood cells. There is a subset of multipotential progenitor cells that have been found to be adipogenic, chondrogenic, osteogenic in their potential. So they can form things like fat, bone, cartilage. And what's very unique about the adipose-derived stem cells is they're angiogenic and vasculogenic in nature. So this is very different than the cells that are found in bone marrow in that they help with that production of blood vessels and vasculature. So some of the current methods used to isolate the adipose-derived stem cells involve using a technique called tumescent liposuction. This is a process that is done under local anesthesia, so the patient remains awake, and you're removing the fat tissue from the subcutaneous space. So it's a minimally invasive procedure that can be done fairly quickly in office. Once you isolate that fat tissue out, we do a washing procedure to help remove any of the red blood cells or the hematopoietic cells that are associated with the population, followed by an enzymatic digestion, which is typically done with a collagenase-based product, and then centrifuging to separate out different layers. And in particular, the layer of interest is called the stromal vascular fraction. The stromal vascular fraction contains a variety of different cells in that population, some of which include mesenchymal stem cells and endothelial and progenitor cells. These are the cells that we referred to in the last slide that are very angiogenic and vasculogenic in nature and can promote healing as well as differentiate into a variety of different tissues. After completing the centrifugation, we isolate that stromal vascular fraction and wash the cells by centrifuging. After that, we do a filtration process to help separate out any fibrous tissue or any chunks of undigested material, followed by preparing the cells in the final suspension for injection. This next slide shows a picture of the separation after centrifuging. So after completing the digestion process with the enzymatic digestion, you can then centrifuge and create multiple layers. If you look at this slide, the top layer is referred to as the liquid fat, followed by the fat below that, and then the media, which is used to separate, which is just the enzymes which are used to digest, and then at the very bottom you have a multiple layer of cells. So at the top you have a white ring, followed by at the bottom a red ring. So this is going to be referred to as your stromal vascular fraction. And this is the fraction that is of most interest. These are going to contain the cells that will be used and can be used for degenerative type diseases or as well as in plastic surgery type applications. Some important considerations when doing these processes within your clinic are that you want to maintain sterility. We feel that it is essential to work underneath what is called a laminar flow hood to avoid any sort of contamination. When you're doing this, this provides sterile air, which is class 100, which means that there are less than 100 particles per million in the environment that you're working in. We validated this process to include working underneath the laminar flow hood, and we use a hood that's actually very realistic for a clinic type setting. It's portable, it's tabletop, it's less than 25 pounds, and so it's something that you can bring out and work under and then put away when you're not using in order to conserve space. The other thing that's essential is doing the washing procedures to remove red blood cells. It has been found that red blood cells can actually prevent the stem cells from doing their job. So it's important to reduce the number of red blood cells associated with the population when offering these treatments to patients. The other thing that's critical is filtering or separating out any of the fibrous materials prior to re-injection. So you want to make sure that you've got a clean sample going back to the patient. And finally, very critical, having a defined and validated cell product. Keep in mind that we are using these products on humans, and so you want to be sure that what you're putting back to the patient is safe and appropriately validated. The next slide shows some of the results from about 60 cc's of fat, and what we typically see is that we are able to isolate about 1 million cells per cc of fat. And when I say cells, this is the mixture. This includes the entire stromal vascular fraction. Within that population, you're going to have a lot of different types of cells. And the way that you can determine which types of cells are in there is by doing something called flow cytometry. And you can look at the protein expression of those cells to determine which population is part of the isolated cells. One of the populations that we look at is CD34 positive cells. These cells have been found to be mesenchymal in nature as well as endothelial and progenitor cells. It's a unique population, in particular ones that are CD34 positive and what's called CD31 negative. These cells, when they're isolated immediately from the body, have the ability to have multiple potential, including things like the ability to differentiate into bone, muscle, cartilage, and to create new blood vessels. So you can see here from this slide that we find typically greater than 50% of the cells in this population are expressing this protein of CD34. This is much higher than what you would typically find from a bone marrow sample. So, for example, from a bone marrow sample, we only get about 1% of the cells in this mesenchymal nature. So we can get up to 500 times more cells that are stem cell type population from fat tissue than we can get from a bone marrow sample. The other thing that's important to look at on this slide is that our viability is always very high. This is because we're dealing with fresh samples. So these samples are immediately isolated from the body and then immediately placed back into the body. If you think about cells, they're kind of like fish out of water. The longer they're away from your body, the less potent and the less viable they are. So the faster that you can get them back to the patient, the better off you're going to be. So when you see high viability, that's because we're dealing with something that is real time. We're putting it immediately back. Just for a reference, the limits, according to the FDA, are that you must have cells that are greater than 70% viable. So we're well above what is acceptable according to FDA regulations. Some of the other things that we typically look for are sterility. We want to ensure that we're not having any sort of breach of the sterility of the product because, in fact, it is going back to patients. You don't want to create an issue. And then also we look at things like endotoxin. These levels are way below what is acceptable limits for delivering back to patients. So let's talk a little bit about the regulatory considerations and what is and is not allowed within your offices today. The FDA currently regulates drugs, devices, and supplements, but the FDA does not regulate the practice of medicine. And most of the things that occur within a doctor's office right now are not FDA regulated or FDA approved. The state medical boards, however,